there's no points, there's no value in figuring out all the specific details and understanding it. If they don't give you points for it, you don't need to know it.
George, welcome back to the MCAT podcast. Are you ready to dive into Blueprint MCAT Diagnostic BioChem Passage 2 today?
I'm so ready. I love the BioChem section. This is like my bread and butter, so I'm excited to get into it.
Why is BioChem bread and butter? Now, obviously, super high test score. You're a Blueprint Live Online instructor, so you obviously mastered the MCAT in one way or another, but why BioChem as bread and butter for you?
I think the same thing with like Chem Phys in some ways. It's that I always felt in cars, like I had to read very carefully and like notice keywords and look at transitions and figure out these big convoluted passages. Whereas in the sciences, it's like, again, it's like this idea of approach. I usually glanced at the passages. Like I would read them, but I wouldn't read them nearly as intently. I would note certain like MCAT testable content. I would note certain relationships, if something increased this or decreased that. I would note maybe certain variables that are mentioned. And I glance at like the figures and tables just to know what's in it, right? But at the same time, you're only ever marked on the questions that they ask you about. You don't need to know every little nitty-gritty detail about these science sections. It's only the questions that they ask you. So if you have kind of a structural idea of what's being presented in each of these bio and like chemistry passages, all you got to do is look at the question, go back, identify if you need a specific detail, or a lot of the questions are actually like pseudo-discretes, which are like very fine bits of content knowledge that they very like minusculely relate back to the passage, as in they'll mention, okay, like they mentioned a protein in the passage and they're like, this property of proteins is such and such. Like it's a discrete question. It's just a content nugget, you know? So that's why I love it. Like I feel like it's very logical, at least for my brain. I thought cars was less logical for my brain, but it took some time and strategy to get there. I've always loved the sciences and kind of the scientific process and like figure analysis. So it was something that really stuck with me.
Yeah. Awesome. All right. So for everyone watching, listening, Blueprint MCAT Diagnostic, everyone gets for free. BlueprintMCAT.com. Sign up for a free account. That's what we've been breaking down here over the last several months, and we're going to continue to do so right now.
Of course. Happy to. So I guess starting with the first paragraph, we'll go paragraph by paragraph, maybe look at like the figure as well. But let's get into it. So giardiasis, or giardiasis, right? MCAT doesn't mark your pronunciation. Giardiasis, caused by the intestinal flagellant protozoa Giardia lamblia, is a common intestinal disease. Giardia are unique in their possession of two nuclei that are similar in appearance, DNA content, and transcription. Usually, G. lamblia are transmitted by contaminated water with cysts. G. lamblia have 7 complex genotypes, but only 2 genotypes cause human infections. The illness is transmitted by differentiation into infective cysts. 3 known cyst wall proteins, CWPs, are highly synthesized in a concerted manner during differentiation into cysts. So looking at this first paragraph, we always ask the class, like, what are you kind of locking into? What are we setting up? What are we going to talk about? And so the big thing that we kind of zone in, okay, we're talking about a certain disease in a certain organism. In terms of highlighting, you might highlight Giardia, like giardiasis or like Giardia lamblia. Doesn't really matter, but just orient yourself to what we're talking about. Something that I really zone into here, though, is like possession of 2 nuclei. I feel like a lot of genetics questions might come down to maybe like ploidy or, chromosomes. So Giardia are unique in their possession of 2 nuclei. I would highlight that. And then this idea of different cyst types, right? So there's 7 genotypes. We have different cysts. So I want to zone in first on this idea of cysts. So transmitted by cysts, and then different genotypes. So again, 2 genotypes specifically cause infection. Maybe we need to highlight, maybe we don't, but we just keep in mind there are different genotypes, and specifically it's the cyst forms that 2 genotypes that lead to these cyst forms that lead to human infections. I also really like to highlight things that have been abbreviated because they're likely going to be mentioned over and over and over. And so in this case, those cyst wall proteins, those CWPs, I might highlight that just to orient myself so that if I see that abbreviation, I'm like, oh, like this is what they're talking about.
Okay.
Amazing. Any questions about like paragraph 1 here?
Nope.
Amazing. So let's dive into paragraph 2, right? So encystation specific vesicles ESVs, give the organism the ability to modify or partition cyst wall cargo during secondary transport. These organelles aid the accumulation and maturation of cyst wall material, CWM, composed of at least 3 cyst wall proteins, CWP1, 2, and 3. So CWP1 through 3, complexed with beta 1,3-N-acetyl D-galactosamine polymer. It's a mouthful. CWM protein is eventually deposited in the plasma membrane of the host's intestinal wall. Trafficking of mature CWM from ESVs to the cell surface, where it forms the cyst wall, is rapid. During cyst formation, a round of DNA replication occurs, then nuclear replication takes place, with the DNA divided equally among the resulting 4 nuclei, followed by a second round of DNA replication. So we get a lot of information here. Again, still a little bit more background. I might highlight ESVs. I see like CWM, just keeping track of new things that are being introduced to. We have this idea of polymer. It's a big, ugly thing. I don't know if I would spend too much time there, but we do get this idea of cyst formation. There's a lot of details about like replication, maybe nuclear division. I'm not going to pay too much attention to it right now. Again, I'm only going to focus on it if they ask me a question, but I might highlight cyst formation just to know that if I do need to come back, I know where to find it. So cyst formation. Now, in terms of the actual figure, here we see, figure 1, regulation of CWP abundance within 2-hour transitions during the 14 incestation time course. Again, I'm looking at this. I wouldn't spend too long on this figure. Again, the idea is, let me get a feel of what the figure is showing me. I'm only going to come back to it if I really need it. So I'm looking at my variables. I'm looking at my axes. I see, HPIE, interesting, percentage of proteins. Somehow it's related. We're looking at some up, down, not changed in my legend. If I need to draw relationships, maybe I'll come back to this figure, but just giving a broad sense of what it might include. Now, for the final paragraph, coming down, right, at any given time throughout the parasite's lifespan, only one CWP gene is expressed. During an infection, however, the particular CWP gene that is transcribed regularly alternates in a process termed antigenic variation. Both expressed and silent CWP genes are located in the nuclear periphery, but in different specific regions. In expressed CWP genes, promoter histone H4 is acetylated on arginine 6 and trimethylated on arginine 2. In contrast, silent CWP genes include a trimethylated histone H4 at arginine 6. During the homologous recombination in cyst nuclei, type 2 topoisomerases assist in chromosome replication and tissue development for Giardia differentiation into dormant cysts. In addition, the level of cyst formation and the CWP1 through 3 gene expression are increased by topo2 overexpression. So again, a lot more going on here, right? So now we're getting an idea of this lifespan. I would say the big thing that I'm locking into here is, again, we're thinking MCAT testable content. Expression, very testable, right? Do we need to know specifics about a parasite's lifespan? Maybe not, but only one CWP gene is expressed at a time. I'm going to keep that in mind, right? I might highlight that. I want to zone in on this infection stage. So I might highlight infection, and I might highlight regularly alternates. This idea that gene, even though one is transcribed at once, we regularly flip-flop. Interesting. And just noting these relationships. And then for the expressed genes, we get later this contrast between the expressed and non-expressed. I might just highlight expressed versus silent. So the expressed and silent genes, just to get an idea of if I need to come back, I can look for those differences. I know it probably has something to do with acetylation or, methylation. We'll look at the specifics. And we're also introduced to this new enzyme, type 2 topoisomerases. If I need to come back to the function, I'll look at that as well. But that's really, again, kind of the structural basis of how I'd approach this passage. And if I need anything, if a question asks me about it, I know exactly where to go back and look for it.
So let's talk about the student who gets to the end and is like, what the heck did I just read?
Yeah.
How is someone supposed to have the confidence that they can move forward to the questions if they have zero clue what they just read?
Absolutely.
Not speaking from experience of like, because I understood this 100%.
Right. But For sure, there are going to be words, there's going to be new things, and this is what I mentioned in like the previous episode of the MCAT is good at dressing things up, core concepts into big, ugly new scenarios, right? You're not expected to know anything about giardiasis. You're not expected to know anything about G. lamellea specifically, right? But they're going to bring in this novel organism to throw you off. They're going to give you, quite frankly, that figure is not like a regular figure that I see often. It's kind of like some sort of weird bar graph looking weird thing, right? They're putting these things in to throw you off. So when you're reading, look for those transition words of like contrast, right? The one contrast I mentioned was the expressed versus silent. If you go in with kind of a structural approach, you can remember where key details are so that if you do need to refer to them, you know where they are, right? If a question doesn't ask you about it, there's no point, there's no value in figuring out all the specific details and understanding it. If they don't give you points for it, you don't need to know it. So in this case, look for what you do know. There's a lot of core concepts like expression, DNA, proteins, how pathways interact. There's a lot of that fits in here. And then relate it back to your core concepts. We might be asked things about expression. We might be asked things about central dogma. Start to anticipate some of the questions you might be asked. And that way you have the confidence that even if you don't understand 100% what's going on, you're not expected to. All you need to know is the same core elements that you've been studying for in your MCAT, and that's enough. Learning the next step to apply it is something that we'll talk about, and you'll see in our practice.
Okay. All right. So we get to the question, and I'll read the question, this first question here, and the answers, and we'll see if we can get to where we need to go. Yeah. So what is the expected ploidy of a cyst that has completed an encystation? 2N, 4N, 8N, or 16N? And I'm like, okay, where did we talk about 2N, 4N, 8N? I don't know, other than like, well, potentially if I don't know anything, is N like nucleus, like nuclei? So I go up here to something we highlighted, 2 nuclei. Okay, so is that I'm going to say this is 2N. This got to be 2N. That's the only thing I see with numbers here. And then we go to incestation, and I'm like, okay, well, we talk about that here. And then the only thing I could find here, right, during cyst formation, we go through DNA replication. I'm like, okay, DNA replication, does that double the number of nuclei? Do we now have 4? Is that 4N? So I want to jump to 4N, but then I'm like, okay, wait, there's more here. Nuclear replication takes place. So DNA replication, nuclear replication, or maybe the nuclear replication is only when it duplicates. And now we're at 4N. DNA divided equally among the resulting 4 nuclei. Oh, it tells me I have 4. Okay, so followed by a second round of DNA replication. Okay, so do I now have 8? So 4 times 2 is 8. So I don't even know if I'm in the right place, but it seems like that's where I'm at. And I think I'm between 8, 16, potentially, maybe, if I don't know exactly what these numbers are in terms of replication, then nuclear replication, then DNA. So I don't know. I'm going to go with 8 because it seems like it's telling me there were 4 and then there's another replication. And so 4 and 4 is 8.
I really like your thought process. And I think the one thing here is that there is a couple of content nuggets that you do need to bring into this question. When we talk about ploidy refers to like the number of nuclei. So like the number of ploidy refers to the number of sets of chromosomes. So if we say 2N, this means a diploid organism. That's just like me and you, eukaryotes, right? We would have 2 sets of chromosome, one from mom, one from dad. So when we have this idea of encystation, just like you said, using our highlights, we zone into cyst formation. I'm going to challenge you that if I tell you that protozoa, so the organisms that we see here, Giardia, it's a protozoa, it's a diploid organism. If it's a diploid organism, that means it has 2 chromosomes, 2 sets of chromosomes per nuclei. We're also told in the passage that Giardia are unique because they start regularly with 2 nuclei. If I give you these content nuggets, would you be willing to try and talk through the process again to see if you can get the answer? I'm not going to tell you if you got it right or wrong. I'm just like, I'm saying, yeah.
So 2 nuclei, both with chromosomes. So that would be 4N to start with. 4 nuclei, 2N each is 8. And then if I double that, then I'm at 16. So I would go with 16N.
16 is the correct answer, right? So your thought process, everything was there. And I really loved that. And so this is why a lot of the students, even when they're like, you know what, they're reflecting, they're reviewing the questions, they're like, I just didn't have the content knowledge. I didn't have the content knowledge. If you had the right thought process, the content knowledge is the easy bit because you can always learn it. You can always memorize it. But the practicing the thought process, even if you don't have the content knowledge, trying to think, what would I do if I had the right content knowledge? That's such an important skill because that's something you can practice on your diagnostic, on your full lengths, even when you don't have all the content areas filled in. So the correct answer in this case would be 16. We start with 2 nuclei, each with 2N. So technically we're at 4N. We do this initial round of DNA replication. So now we're at 4N each in our 2 nuclei. So we're at 8N. We do this nuclear division. So we're just breaking apart the sacs into 4 nuclei instead of 2. That 4N gets split evenly into 2N, 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 2N. So we're still at 8N. And then the second round of DNA replication, all those 2Ns become 4N. So in the end, we have 4 nuclei with 4N each. The total ploidy would be 16N.
Yep. Got it. Simple math once you understand the concept behind it.
Yeah.
Got it. All right. Why don't you read the second question here and I'll try to.
Amazing. I like this one a lot. So the functions of the ESVs of Giardia most closely resemble what cell organelle? Lysosomes, A. B. Golgi apparatus. C. Nucleolus. Or D. Centriole.
Oh, man. So going back to some basic cell biology here. So the we're told here that ESVs, right? We highlighted ESVs, so we can go right to that.
Excellent.
Give the organism the ability to modify or partition cyst wall cargo during secretory transport. And so I'm like, okay, transport. What is involved in transport? That's what I come to. And so I'm trying to remember what is involved in transport. And I go, well, nucleolus, I don't think is involved in transport at all. That doesn't ring any sort of bell. Centriole, I'm like, okay, centriole, that's kind of hanging out with the nucleus, doing stuff. That doesn't seem right. Then I get to lysosome. Well, I should have started at lysosome because it's the top one there. Lysosomes, to me, I'm like, oh, that's just what like breaks down everything, right? That just goes and breaks down everything. I'm like, well, is that transport? Is that what that is? And then the Golgi apparatus, I'm like, well, the Golgi apparatus is like, that's where it's packing stuff and transporting stuff. That's like the Amazon warehouse of the cell. I'm like, well, that's transport. It's logistics. It's like UPS. So I'm gonna go with Golgi apparatus just because like I think that's where we're at.
Couldn't have said it better myself, right? That's the correct answer. B is the correct answer. It's the Golgi apparatus. And with the kind of approach to this question, you did it perfectly. We see the function of ESVs. Okay, well, we do need some sort of passage information. We're not expected to know ESVs or Giardia to begin with. So where is it? It's probably in the passage. We look at our highlighting. It mentions, okay, it's the ability to modify or partition cell wall cargo during secretory transport. If you paraphrase this is just like packing, modifying, changing as you send things off. The whole function of the Golgi apparatus, just like you said, it's the packing and sorting center. It's the place where you add things and remove things. It's like the Amazon. I always thought of it like the Canada Post, but like same kind of thing, right? So I'm up in the north, like Canada Post, right? But it's the packing and sorting center. So that's absolutely correct. For the sake of completeness, lysosomes, just like you said, they break things down. They're little packets of like very, destructive things. Think of it that way. The name itself, lysosome, lysis means to break down. Makes sense. Nucleolus, it's a center in the nucleus itself. It's really responsible for kind of like rRNA transcription and processing, making those ribosomes. And then centrioles, those are like the fishing reels of the cell. When you're dividing, they're at the opposite poles, and then the microtubules attach and things move along them. So excellent. B would be the correct answer here.
Whew. All right.
How you feeling?
God, stressed. I'm thinking about it, those negative podcast reviews going, Dr. Gray doesn't know anything. He doesn't help at all.
They're yelling, the answer's B. The answer's B.
Question 8. Given passage information, okay, so they want us to go to the passage, Giardia most likely transports which of the following proteins external to the organism itself? A, CWP3, B, histone H4, C, beta-1,3-N acetyl D-galactosamine, or D, ESV. All right. So ESV are interesting because I'm like, well, that doesn't seem right, because we were just talking about ESVs as like the thing that partitions and transports and these organelles aid accumulation and maturation of cyst wall material. Composed of these things that go into it, which CW3, 1 through 3, which is answer choice A, complexed with the beta-1,3-N acetyl D, whatever, is answer choice C. I'm like, well, if A and C are packaged together, they both can't be right. Or one of them can't be right, and one of them can be wrong, potentially, as I'm reading that. So I'm like, just by process of elimination, A, C, and D are all kind of related. So I'm going to go with B just based on that.
Fair enough. It's a good thought process. And like that process of elimination is really useful for answering questions. Unfortunately, the correct answer in this case would actually be A. And so let's talk about it, right? Yeah. I think using the same kind of logic, you're absolutely correct. If you're not sure, look at where we talk about transport. There's one line and you were almost there. The next line right there says CWM protein is eventually deposited in the plasma membrane. This is the part that we need to focus on, because when we talk about which of the following proteins would go external to the organism, the only way it goes external to the organism is if it passes everything and ends up at the membrane so that it fuses and is shunted out. Yeah. So we need to think of something that goes to the membrane, right? And we look at CWM. CWM is not in our answer choices, but we know that CWM is made up of this CWP1 and 3 proteins. Yeah. It might be tempting to pick this this N-acetyl-galactosamine thing, but this is part of the cyst wall. So it's kind of like, it's a layer just within. It's not quite at the membrane. It's a layer just within. So if you think about it, it would actually be flipped in the bottom. It would never make it out to the extracellular space. In terms of ESVs, your thought process was perfect. We just talked about it. It's the packing and sorting center. It can't actually be shunted out. Histones. If we think about histones, what do they bind? They like to bind DNA, right? They like to bind, the histone modifications can cause like winding or unwinding of DNA. Histones would be located in our nucleus, right? Yep. If they're located in our nucleus, they wouldn't be shunted out. But excellent approach to this question. Yeah. And
it literally is very like right here, trafficking of this to the cell surface. Blah blah blah. All right, bummer. I tried to hack that one because I'm like, okay,
it's a really good strategy. Yeah.
Right. The stress of the MCAT is like, I need to go fast. So I tried to shortcut that, going, let me see if it works.
Yeah,
it didn't.
But definitely, there's a ton of questions that there's always an easy way. Always check to see if there's an easy way if you're not sure.
Yeah. All right, go ahead and read 9.
Yes, absolutely. So question 9. With which of the following do the arginine side chains found on histone proteins most likely have the strongest interactions? A, thymine groups on DNA. B, adenine groups on DNA. C, phosphate groups on DNA. D, oxyribose on RNA.
Which of the following do the arginine side chains found on histone proteins most likely interact? So you just mentioned histone and DNA. So I'm going to steal a little bit of that knowledge and get rid of answer choice D right off the bat, because I'm like, oh, well, that's the one that's unlike the other. So thiamine, adenine, phosphate. So it's interesting. Two of these are kind of our DNA, right? If we think of CGAT kind of stuff. But phosphate groups are different. And I'm like, well, again, I'm trying to shortcut some knowledge. Can I just look at these and go, I don't really know, but can I make some sort of educated guess? Do I remember anything about histone liking A or T, thiamine or adenine too much? Well, A and T are going to be kind of together all the time because that's what pairs together. So that seems weird. So I'm going to go with C just because of that.
Flawless logic and excellent execution of strategy, right? The correct answer is C. And I really love the way that you zoned in on T and A, like the bases, thiamine and adenine, because you're absolutely correct. This is one of those cases where because it's kind of like the same thing, it can't be anything unique. If you have two things locked together, they can't both be true. Yeah. In this case, the actual science behind it, I always like to look at questions and paraphrase it into something simpler, right? Which of the following would arginine side chains, on histone proteins most likely interact? Briefly, this just means like, what interaction is most favorable with arginine, right? And if I ask myself arginine, if I think back to my amino acids, arginine is one of those basic amino acids, which means at physiologic pH, it's going to be positively charged. What's a favorable interaction? The positive charge wants to interact with negative charges. So if I use that approach, I could say, okay, well, I look at my list, thiamine, adenine, phosphates, and oxyribose. Phosphates, if I remember like the structure of phosphates or remember what they roughly do, super electronegative. They often have like, their deprotonated form. So that's a negative charge. It wants to be next to a negative charge. Phosphate's the correct answer. But I love the way that you eliminated based on the RNA. Histones can mostly, especially in this case, if they're in like the nucleus, they're going to be associated with DNA. You also eliminated T and A simply because like, that they're going to be together, and it's pretty much an equivalent answer. So you could also use process of elimination to get the fact that C would be the correct answer, phosphates.
All right. I did it. All right. Question 10. The data in the passage suggests that which time period during incestation is the level of protein dropping most quickly? A, 0 to 2, HPIE, hippie. 4 to 6, HPIE, 10 to 12, HPIE, or 12 to 14, HPIE. So we go to our graph, right? Because it says the data in the passage, and this is where our data is displayed here. And now I need to figure out what's going on. And so if I look at percentage of proteins here on the left, I see HPIE here. I see the legend. The darker one is going up, I'm assuming. But it wants us to know dropping down, potentially, if I read that correctly. Maybe. And we see 4 to 6 has the biggest going down. And so I just want to double check, am I reading that right? Or is it dropping most quickly is up? So let me see. Let me read here. Regulation of CWP abundance within 2-hour transition during the 14-hour incestation time course, HPIE, hours post initiation of incestations, N equals total protein in sample. So total protein in sample, if we look at dropping, so level of protein. So if N, let's look at this. If N is the total protein in the sample, do we look at just the ends going from number to number to number going, okay, which number drops the most? If I were to do that, it would be 6 to 8. So now I'm confused. Is it 4 to 6 or 6 to 8? 6 to 8 isn't an answer, so I'm going to go 4 to 6.
You worried me a little second with your final analysis there. So, but you're absolutely correct, right? And so sample size, just to clarify, N is always like the amount of things that you're going to look at. So what's in your group when you make these conclusions? N itself doesn't really tell you anything about results. It just says like, I did this study with 500 cysts, right? The N sample is you worked on 500 cysts, tells you nothing about what changed within the group. So in this case, we're talking the data in the passage, looking at which time period had the biggest drop in protein. I loved your approach. You went straight to the figure because this is the results. When we want results, we're going to look at data. We're going to look at figures. We came to this. And the other clue too is like this hippie thing, HPIE. Where did we see it? We only saw it in the figure. So go straight to the figure. In this case, just like you said, when we pay attention to our axes, percentage of proteins, okay, HPIE, we look at our legend, up, not changed, and down regulated, we see that in the 4 to 6 group, it has the highest amount of down regulation. We also look at our figure caption and it says regulation of CWP abundance. So if we have more down regulation, the percentage of proteins that are down regulated, if it's more down regulated, you have less abundance, therefore less proteins expressed. This is also another instance where you could use like which one is not like the others, right? If you look at C and D, so we look at the 10 to 12 and the 12 to 14, how would you describe those? Are they like pretty
equivalent?
Different?
Yeah.
They're pretty much the same, right? So same kind of idea. You can get rid of both of them because they can't both be right. And then we look between 0 and 2 and 4 to 6. 0 and 2 had an up regulation. There would be more protein, whereas 4 to 6 had less down regulation. Amazing.
So again, I think the whole point of this podcast and how we are breaking down these questions is really a show. Like, it's been a long time since I've studied this material. I don't come into these looking at the questions beforehand. I truly am just kind of going on the fly. And I'm sorry if that's frustrating for some of you that I don't remember some of the basic biology here. But I think the goal is to show that even without this information, I can still get to the right answer. This test is about thinking and analyzing, not about knowing. So hopefully this is helpful. George, as you get to the end of a passage as we are here, what are your recommendations for the end of passage, beginning of next passage kind of transition?
Honestly, it can be as simple as like closing your eyes, taking like a 5-second breather, leave everything behind that you just read. You can't take it. It doesn't transfer, right? In the sciences, actually, that's the one thing is in the sciences, you might have some content knowledge given to you in a passage that you can use later. But for the most part, try not to worry too much about the last passage. Take a breath. If you flag some questions that you know if you had more time for, you could work on, keep them in the back of your mind. If you have more time, you'll come back, but make sure you answer every question. Never leave a question blank, because even if it's a guessed correct answer, you still get the point. You get no penalties for getting it wrong. So answer every question, take a deep breath, take that leap of faith, and start the next passage with a fresh mind and approach.